dois, três, quatro, cinco, seis, sete, oito, nove. Dois, três, quatro. Testing, testing. Testing, testing. One, two. Tá?
Finding your seats. We're just about to get started with our next session. All right, thanks, folks. Okay, as you're getting settled in, we're about to start our next session on driving collaboration in neurophysiology with NWB and DANDI and IBL. Yeah. All right, great. So join me in welcoming up to the podium Ben Dichter and Ryan Lee.
Hi, everyone. I'm Ben Dichter, and Ryan Lee will be presenting with me on driving collaboration in neurophysiology with NWB and DANDI. This will be a two-parter, so after this, the IBL team will present their work as well. This is, I have here on the right the leads of the NWB and DANDI projects. I'll say we're going to have a lot of content in these slides. A lot of these slides have links as well that you can use to dig in further. So I would really encourage you to go to this URL, bit.ly/NWB-Cosyne-2026, which leads you to the Google Slides of this presentation. And yeah, you'll be able to follow all those links and dig into this more thoroughly. It's really an honor for us to be here. We're representatives of a much larger team that's been working on this project for 10 plus years across many different institutions. So I just want to acknowledge that we're representatives of a much larger team, and it's really great to present here at Cosyne to all of you. So first, let's talk about our vision of why we actually need a data standard and archive for neurophysiology. So first of all, neurophysiology data is very expensive to collect. It's expensive in terms of money, the very expensive equipment that we use, in time, both in the many hours and months of time of highly trained people, and also the amount of time it takes to actually get a study out and into publication. And then we also use animal lives, which I think means it's really our responsibility as scientists to learn as much as we can from the data that we collect. And we know that the data we can collect for one study can be used to answer many different scientific questions. But it can be really difficult to share data with others. in our experience talking with labs, it can be very tedious and difficult to share data even within a single lab. And sharing data with collaborators in another lab or another institution can be even harder. Everyone basically has their own way of storing data. There are dozens of different acquisition systems that have dozens of different data formats. The metadata is distributed across often different computers, sometimes in lab notebooks, sometimes in Excel spreadsheets in all different types of formats. Sometimes it's missing. Because of this, it can take weeks or months of back and forth communication for one lab to be able to use another lab's data set. And on top of that, it's difficult to share scientific software. Often, using software requires converting data from one format to another, so you have all of the problems of understanding the data format. And often the metadata is inconsistent, and it can sometimes be impossible to go to the desired data format in order to use a software tool. So as a result, without data sharing infrastructure, scientists often just collect new data and write new software tools when they don't need to. They could have used someone else's work. They could have built upon it. And we think this is really a big problem in the field, honestly. This is really bad for our efficiency if we have to keep on reproducing work, redoing things that have already been done. It's really bad for quality because you can make mistakes if you keep on doing it over and over again, whereas you can find mistakes if you keep on reusing things. And it's bad for reproducibility. And we have a vision for where we want the field to move, and we've been working hard to try to enable this vision. And that can basically be summed up by when a neuroscientist has a scientific question, instead of thinking, what experiment should I run to answer this question, they instead think, what query should I run on open data? What data can I find that can help me get to the next step of this question? Maybe a data set that would work for me already exists. Maybe it doesn't, and then you'll need to collect the data. But it's worth at least trying to look. And I hope to convince you here that we have the resources to make this not only possible, but really seamless. And that's what we've been working on for the last 10 years. And on top of all of that, funders and journals are increasingly requiring data sharing. So we're seeing announcements by government funders like the NIH, like from foundations like the Michael J. Fox Foundation with the Aligning Science Across Parkinson's, like from journals, like Nature has released a statement. All of these different organizations, all of these quotes basically say you have to share your data in different ways. But that's basically the message across all of these different organizations. So the outline of this talk is first we're going to tell you about the data standard, NWB, and then we are going to tell you about DANDI, the data archive that you can use for sharing this data. And then we'll walk you through how you would find and analyze NWB data on DANDI to answer your own scientific questions. So the two main underpinning technologies that we're going to present here are Neurodata Without Borders, or NWB, and the DANDI archive. So NWB is a standard for storing and analyzing neurophysiology and associated behavioral data. And DANDI is an archive for sharing and analyzing neurophysiology and behavioral data. So I like to think about this as NWB is kind of like an MP4 that provides a common format that everybody can distribute and many different types of software can read. And then DANDI is like YouTube that allows you to upload, download, stream data, share it with anyone in the world for free. So first we'll go into Introduction to NWB, and Ryan Lee will take it from here.
Great. Thanks, Ben. All right, so what do we mean by neurophysiology? Well, we define it as the study of neural activity at cellular and circuit resolution. And this includes electrophysiology, such as neuropixels and patch clamp recordings. It includes optical physiology, such as calcium imaging, voltage imaging, and fiber photometry. And it also includes behavior, such as the video and audio that are often recorded alongside these neural data. And in each of these cases, you have the raw data. You have the raw voltage recordings, the images, and the video. And you also have the results of critical processing steps, such as spike sorting, image segmentation, and pose estimation, that transform your raw data into a form that's ready for analysis. And in NWB, you can store both the raw and the processed data. And we encourage you to store both so that future you and others can reproduce and reuse your work. Now, raw and processed data alone isn't enough to be understandable and reusable. The goal of NWB is ultimately to store all of this neural and behavioral data together with all of the metadata necessary to understand and analyze it. NWB defines a unified data format for the most common types of neurophysiology, the data and metadata, including the neural and behavioral data I just mentioned, and critically, also metadata about the data acquisition, such as the device settings and the brain area targeted, information about the experimental design, such as the stimuli and the trial structure, and information about the subject, such as its species, genotype, and age. Now, if you look under the hood of an NWB file, you'll find a hierarchical organization of data and metadata stored in specific places. Here's an example of an NWB file containing extracellular electrophysiology data. In the top level general group, you can see information about the experimental subject, the experimenters, and information about the electrodes, such as their locations. In the acquisition group, you can find all of your raw data. Here we have raw voltage data stored in an electrical series data type, and this includes a 2D array that's timed by electrodes, the timestamps for each sample, and the electrodes axis, which links to the table of electrode metadata that I just showed in the general group. And this data type also includes metadata needed to interpret the array, such as the units of measurement, the sampling rates, and any conversion factors. You'll find process data in the processing group or the units table. The units table contains information about any spike-sorted units, including the spike times, the mean waveforms, and other features of each unit, and which electrodes each unit was observed in, which again link back to the electrodes table. All of the data and metadata for a single experimental session are stored in a single NWB file. And that NWB file can be written with our HDF5 or ZAR backends, depending on your needs. Now, this standard organization is great for data types commonly used in the field, but as we know, neurophysiology is constantly innovating new methods. How do we ensure those specialized and new data types that are not supported by the core standard can be stored and shared as well? In those cases, you can create an NWB extension to describe your new data type and metadata for it. And this is particularly useful for when you want to store data and metadata from a new method for acquiring or processing data, or you want to store metadata that's specific to your lab or your experiments. If you use an extension, it's packaged with the data so that other people and software tools can understand what your new data type is and how it's structured. You can find over 30 extensions developed by the community in our NDX catalog, and you can develop and share your own extensions in this catalog as well. Okay, so that describes kind of at a high level what the NWB standard is and how it can support the most common neurophysiology data types and metadata, as well as be extended for other types not supported. How do we get data into the standard? You know, neurophysiology experiments, as you know, are quite diverse. They generate a lot of data and often involve combining data from several different modalities. So you might have a NeuroPixels recording from a Spike GLX system. You might have the sorted units from that recording from kilosort in a 5 format. You might have raw video data in MP4, and then pose estimates from that video, the stimuli, et cetera. How do you get all of this data into NWB? Well, we provide 3 sets of tools at different levels of accessibility and flexibility. First, we have NWB Guide. This is a desktop app that provides a graphical user interface for you to fill in the details about your experiments and where your data files live. It supports over 40 common data formats and will validate your metadata as you enter it. And it can batch convert all of your data from all of your sessions to NWB and shows you interactive visualizations of your converted data so that you can inspect it and verify that everything is correct. And finally, you can use Guide to upload your NWB files to the DANDI archive, which we'll talk about shortly. And you can do all of this in Guide without having to write a single line of code. Now, Guide works for converting data from certain supported data types to NWB, many of them. But if it doesn't support your data formats or you want to convert data programmatically, we recommend that you try NeuroConv. NeuroConv is a Python package that converts data from more formats to NWB and provides more options for customizing how it's converted. Conversions generally require a small amount of Python code. So in this example here, we're converting a single Spike GLX recording to NWB using NeuroConv, and this requires about 10 lines of code to import the packages, locate the source data, customize the metadata, and then run the conversion. And here is a full list of all of the different data formats currently accepted by NeuroConv for conversion to NWB. If your favorite format isn't on the list, please let us know, or create an issue on the NeuroConv GitHub repo, and we can work with you to support it. Finally, we provide APIs in Python and MATLAB that allow you to read and write NWB data. And these are useful if you're working with data in a format that's not supported by Guide or NeuroConv, such as a custom MATLAB file, or maybe you have trial information in a custom Excel spreadsheet, or you're using extensions, or you just want fine control over how the data is laid out on disk. We have a ton of documentation and tutorials for how to use these tools, these APIs, at these links. And these tutorials range from the basics of reading and writing different types of neurophysiology data, specialized tutorials for ePhys, for calcium imaging, etc. And it extends to advanced topics such as customizing compression and streaming data from the internet. We're also excited to show off our new conversion chatbot that's integrated with PyNWB and NeuroConv documentation. So let's just go to one of those pages now. Let's hope this works. This is the PyNWB docs. We'll just zoom in. And we can open the assistant. It's just a little pop-up on the right. And ask it, help me convert Spike GLX and Phi data to NWB. And if the internet's working, then good. It will go through all the docs and report back some code snippets on instructing you exactly how to do this for your specific data types, including citations back to the source pages for these documentation. Yep. So we're excited about this handy feature. And we'd love your feedback on it. So if you try to use it, please let us know. We're also developing ACK-NWB. This is a C++ library for recording data directly to NWB. And we're working with data acquisition systems like OpenEFIS and BlackRock to integrate this with their systems so that you can get NWB files straight from the box without needing to convert your data after acquisition. So, as you can see, NWB is much more than just a standard. NWB is also an ecosystem of software tools that work seamlessly together to make it easy for scientists and developers like all of you to work with NWB data. There are core tools for converting data to NWB and validating data, such as Guide and NeuroConv that I just described, as well as the inspector for enhanced validation. I've told you about our APIs in Python, MATLAB, and C++ that allow you to easily and efficiently read and write NWB files. There's also tools that help develop and share NWB extensions. And then there's other low-level tools that support the standard and the software. We, the NWB team, we develop and maintain all these core NWB tools. And these core tools support a vast and growing software ecosystem of over 30 community-developed tools that can read and or write NWB files. And because they share NWB as a common format, these tools can be chained together into a data analysis pipeline that's tailored to your needs. And this ecosystem includes tools for data exploration and visualization, such as NeuroSift, which Ben will demonstrate later, tools for processing and analyzing specific types of data, like for extracellular ePhys, Spike Interface accepts NWB as a common input and output format, and provides access to a wide array of spike sorters. We also have tools for intracellular ePhys, for optical physiology. You might be familiar with Suite 2P or Cayman, and for behaviors, such as converting outputs of deep lab cut and sleep. Finally, we also have tools for general analysis and data management that work well with NWB. And you can learn more about all of the various tools in this vast and growing ecosystem at nwb.org/tools/core or /community. And finally, to round off the NWB section, I want to emphasize that NWB is not a static data standard. It continuously evolves to meet the needs of the community through a community-driven process. Major changes such as integrating extensions into the core standard will go through an NWB enhancement proposal process, where proposals are submitted, reviewed by the Technical Advisory Board, and evaluated by the broader community. And this keeps NWB current with the latest methods and data types in neuroscience. We have 4 proposals currently active. Proposal 1 has been accepted and is being integrated into the core software, and Proposal 4 is under review. And as part of Proposal 1, we're adding support for annotating events with hierarchical event descriptors, or head tags, which is a controlled vocabulary for describing experimental events. And this allows scientists to describe their stimulus and behavior data more formally, and enables the analysis and visualization tools in the ecosystem to better interpret and process these data. And we're big fans of integrating across data standards and tools across the community, and this is just one example of that. All right, now I'll pass it back to Ben to tell you all about the DANDI archive.
All right, thanks, Ryan. Okay, so let's say you've converted your data to NWB, or you want to search for existing NWB data for your project. That's where you would turn to the DANDI archive. So first, about contributing data to the DANDI archive. So DANDI stores neurophysiology and behavioral data in the NWB format. This allows it to validate all of the NWB data. It checks that data for best practices, and it automatically extracts metadata, which then allows it to expose search features over all of the NWB data that are deposited. You can upload hundreds of terabytes of data to DANDI for free, neurophysiology data. This is because of a partnership with an AWS Open Data Sponsorship Program. Basically, Amazon allows us to upload, download, and store data on S3 for free, and so now all of you can use it for free too. It can handle, basically with this allocation, we can handle all raw data from most neurophysiology experiments. And I'll show you, we actually have the raw data from the massive IBL data set that we just put up that you'll learn more about, as well as data from the Allen Institute and many others. We would just ask that you talk to us if you want to store something over 10 terabytes. We definitely do accept data at that size. We just want to make sure that it's appropriate for the archive. It has all of the lossless compression optimizations that would make it optimal for the archive. But just please come by the booth or just connect with us online and discuss. You can put data up publicly or in embargo mode, which essentially allows you and only you and the co-owners to view the data for a limited time. The licenses that we support are open licenses, CC0 and CC BY, which basically means all of you can use all of the data on the archive for free. Actually getting data up, there are 2 methods here. One is using NWB Guide, which Ryan presented. That's just the application. So that's like you double click it, you can drag and drop your data in, and then it'll walk you through uploading it in a graphical interface. That's the easier method. We also have a DANDI command line interface, which is a little bit more flexible and powerful. It does require you to be familiar with the command line and comfortable with that. So this is a summary of all the data. This is actually a plot that I'm really proud of. This is all of the data on DANDI, almost 500. We have 489 data sets now published on the DANDI archive in NWB, and we are seeing super linear growth. So we're just really excited to see how this will expand even going forward into the future. Over 200 of these data sets are of mice. We also have lots of rat and monkey and human and zebrafish, the common model organisms, but we also have lots of other species as well. We have fruit flies and hamsters, and we even have one on ants. So if you're working with a less common model organism, we'd be happy to talk and support your data as well. We have over 200 extracellular electrophysiology data sets, over 100 behavior and microscopy such as calcium imaging, and we have over 50 intracellular electrophysiology experiments. So just a massive volume of data here. Just a few to highlight. We have the microns 2-photon functional imaging data set. This is 1.3 terabytes of optical physiology data for a visual task. The really cool thing about this data set is they took that tissue and they did electron microscopy on it, and then they're co-registering the structure, the cells that they find in the EM with the 2P data. So you can actually compare the functional and structural connectivity of these neurons. The IBL brain-wide map, which you'll learn about over the next hour, is 45 terabytes. We just recently released this like a few days ago. It has neuropixel, eye tracking, video, and visual decision-making data. And the Allen Institute has deposited over 30 data sets on the archive. Honestly, the Allen Institute has been one of the biggest supporters of NWB from day one. This includes data from BICAN and BICCN, from the visual coding experiments, from the SHIELD project, from the OpenScope project. And one really cool thing about the OpenScope project is why some of my favorite data on the archive is it also has an OpenScope data book, which is an executable notebook that shows you how to understand each of the OpenScope data sets, how to run common data visualizations and analyses, and even goes into more advanced stuff like how to analyze with Zebra. And this is all executable, and they even like provide environments to make sure that this runs. And you can see this just like this massive textbook that they've been building over years that works seamlessly with the OpenScope project data. So if you're kind of new to this area, this is like one of the really, or even if you're not new to this area, you've got a lot to learn from here. Now, these are just a few of the hundreds of data sets in the archive. It's really hard to summarize everything, so I just made a word cloud of all of the descriptions of the data sets. So hopefully you can kind of get a sense of all the data on there, but it's really quite diverse. So we also, in DANDI, provide DANDI Hub, which is a free cloud compute resource that gives you fast access to data on DANDI. So you can download or stream data directly to the DANDI Hub. We have different server options, including a GPU option, also for free. Please don't use that to mine crypto, just for data analysis. Along with the DANDI Hub, we have this repo for example notebooks. So this allows you to find and share analysis notebooks. We already have dozens of notebooks that point to specific data sets. You're welcome to search that and use that as a launching point for your own analysis, or to share a notebook that you've created on a specific data set with the community. And then that will be runnable on the hub and also on any other compute resource. Okay, now finally I want to talk about finding and analyzing NWB data on DANDI. So searching DANDI is as simple as going to dandiarchive.org and using the search bar. That's the simplest way. If you type in neuropixel, you'll find 47 data sets that mention neuropixels. If you type in hippocampus, you'll find, I think, over 80 data sets. So that's like pretty basic, but you might be able to just get rolling with that. We also provide much more advanced search tools where you can programmatically search. So let's say if you wanted like a mouse data set with at least 3 subjects that had at least 2 sessions of at least an hour, and it had this many units or something like that, you can construct a very complicated query in Python and search over the whole archive. We have linked to tutorials here for like more basic versions of that, and then really intricate versions of that where you actually inspect the NWB files themselves to get really a lot of metadata. So you're welcome to check those out if you're interested in that, or just talk to us and we'll walk you through how you would find what you're looking for. Another way to explore the archive is a new tool that we just released called atlas.dandiarchive.org, which I have pulled up in this tab, which you can see. Great. So this is an interactive tool for exploring the mouse CCF data on the archive. So I'm visualizing the Allen Institute CCF Atlas here, and we've indexed 45 different data sets that have sufficient metadata within NWB where we can actually map them to this common atlas and display them all together. So I'm just going to go to a brain area. And here on the right, you can see all of the data sets that, that have recorded from that brain area. So I'll just go to the IBL and select a session, and then here you can see the neuropixel probes that were recorded in this session. So I'm just going to X out of this because I happen to know that 1051 is a really good one. Okay, so this is the SHIELD project from the Allen Institute, where they really made advanced technology for surgical implantation of neuropixel probes. And yeah, you can see many different, so this is a single subject, multiple sessions. They really get a great sampling of data here. So you can explore what data is available with this atlas, which is live now at atlas.dandiarchive.org. And then you can dig into those specific sessions by downloading that data. Okay, so let's say you found a data set that you're interested in, maybe with the atlas, maybe with search. Now you want to start to visualize and explore that data. And that's where NeuroSift is a really powerful tool. So this is a tool built by Jeremy Magland at the Flatiron Institute that provides interactive visualizations of NWB files. So basically it can understand the structure of NWB, it can stream data directly from the DANDI archive, and it runs directly in browser. So there's no need to install anything, and you can just, any URL that you get, you can just pass to your colleague, and then they can do the same. So I'm gonna show you two demos of this. One is on the IBL data set. So I'm just gonna first show you how I would actually get there. So here's dandiarchive.org. I'm just going to type in IBL, and I'll use this IBL brain-wide map. I'll go, this is, by the way, this is a DandiSet landing page. This has all of the metadata about the data set and provides a button to go and view the data itself. I'm just going to go to the first subject, first session, open with NeuroSift. Now, on this, I really like the PSTH option, and I'll change the settings to Gabor stimulus onset time for the alignment and the feedback time, and view all units. And then I'll group by the Gabor stimulus side. So here, this is streaming data live from the archive and generating these PSTHs on the fly. And if I scroll down, you can start to see really interesting response patterns. I particularly like unit 26. I do, thank you. Onset time feedback. There we go, thank you. So, so this unit I thought was really interesting because it has this strong response that responds more to the left visual stimuli than the right. Where the neuron increases activity, but then on the feedback time, it has this even stronger decrease of activity. So, so I just think, wow, like, what's this neuron doing? Like, what's it representing? How is it used in the task? I feel like these PSTHs, when you really start to look at them, you can really just start to generate hypotheses about how you'd want to analyze that data. So, so there's just thousands of these neurons from all these different brain areas, and it's really fun to just look through the data like this. I mean, the NeuroSift works on every data set, NWB data set on the archive. Another really cool one is this data set from the Moser lab that was one of the key data sets that was cited in their 2014 Nobel Prize. So if I go to this DandiSet landing page, I'll go to Files, I'll just go First Subject First Session, and I'll show you that NeuroSift can do multiple visualizations simultaneously. So let's look at the electrical series LFP, a raster of the units, and then two visualizations for the spatial series. And then I'll zoom in a little bit. All right, great. So this is the LFP data. This is a raster of the spikes. This is X and Y over time, and this is X versus Y. And all of these views are synchronized with each other, which allows you to sift, sift through the data and really inspect this data, understand what data sets would be relevant for you. Now, finally, let's say you maybe use the atlas, maybe you use Neurosift, and maybe you use the search bar. You've identified a data set that you want to analyze. And now you're off to try to analyze it. One of the most powerful tools that we integrate with is Pineapple, which is built by Guillaume Viejo also at the Flatiron Institute. So the Pineapple basically works by, it takes this input data from various different modalities and represents them as different types of objects. So time series, interval sets, timestamps. And then once it has those different types of objects, it can define methods over those objects that are common methods that you need to do in neurophysiology, which enable common analyses like peri-event histograms, tuning curves, decoding, cross-correlation. So now I'm going to show you a demo of this Moser Lab data set that leverages all of these tools. So I'm going into here, and if you want to analyze this data, you can go into Python usage, which shows you a script that shows you how to stream this data directly to a Python session. So this is just a local Python session that I'm going to paste this in. So, so here it's using Lindy, which is a technology for streaming data from the archive, and then it's showing how to read basically every different data element in the NWB file. So if I just type NWB, you'll see navigation around that. You can go to any one of these, so LFP electrical series.rate, for instance. And there you go. We're streaming this data live from the archive. Now, I'm going to analyze this with Pineapple. So import Pineapple as nap, import matplotlib. Oh yeah. Okay. Now, is that visible? Can I get a thumbs up, thumbs down in the back? A reluctant thumbs up. I'll accept that. Okay. And then let's use Pineapple to read the NWB file. So that's nap.nwb file NWB. Nap NWB equals. Okay, so this goes into that NWB file and finds all of the data streams that can be represented as Pineapple objects. Now we can use the Pineapple analyses, nap.compute tuning curves, nap NWB units, nap NWB. I don't trust myself to type this. Bins equals 20, and we'll call this rate maps. Okay, and now for x in rate maps, plt.figure, plt.imShow x, plt.color bar, label equals firing rate hertz. Okay, and there we go. Rate maps demonstrating grid cells in the medial entorhinal cortex from the Moser Lab. So Nobel Prize speed run, I guess. So it's really that simple to stream all of these together. And just to reiterate, this is really made possible because a number of different technologies working together well. The open data from the Moser Lab, obviously very important. The NWB data standard, so this can be represented in a way that tools can understand it. The DANDI archive making this data accessible. Neurosift making it, giving us the code for streaming. Lindy for the streaming layer, and Pineapple for analysis. And then, of course, this is built upon the open Python data science stack as well, like Matplotlib and NumPy and all the rest. All of this works together to make this possible. So in just a few minutes, you can get these rate maps. And here's the code for later if you want to play around with it. Now, we've seen many groups using open data in their publications. And this is really the next step for us, is really trying to encourage this as much as possible, because now we have so much valuable data and tools that enable this. We have now, last we counted, 168 reuse publications, though we're still finding more every day. And some of these are really high-profile publications. This paper on dentate spikes in Nature recently heavily used open data. The Zebra paper, which was presented here at Cosyne recently, was this panel here was created from NWB data that they took directly from the DANDI archive. The KiloSort 4 paper for spike sorting, several of the data sets they tested on and demonstrated the performance of their algorithm were from DANDI and NWB. And this POYO paper is a paper on training a neural foundation model that used several data sets on the DANDI archive. So these demonstrate different types of ways that people are using the NWB data in influential papers, sometimes to discover new scientific findings, sometimes to build new powerful tools, which can enable hundreds of other labs, and sometimes to build new machine learning models, and all of the above. So we want to support as many different types of use cases to accelerate science. One thing I will say is if you do use data, one thing we're trying to encourage is that people cite the data sets themselves, like the DOIs of the data sets. Also publish the paper associated with the data, but the thing is if you publish the data set, then we can start to track it and then we can understand when people are reusing data. Without mentioning the DOI of the data set itself, it's much more difficult to track. So one really amazing thing about this project is we've been able to see people using this data all over the world. So it's not just in the US and Europe. We're seeing lots of people use data in South America. We're seeing all over Asia, in Oceania, and you know, even in Africa, it looks like there's someone in Madagascar. We're very curious who is using DANDI in Madagascar. And yeah, we're just really excited to see this world map grow and to really to embody the name of Neurodata Without Borders. This is exactly what we want to see. So I'll just throw up a bunch of links here about how to get in touch with us, ask us questions on the help desk, connect with us on Slack. We're happy to do one-on-one consultations. Please sign up for our newsletter if you're interested in learning about any of our events where you can learn much more in depth how to convert or analyze data. Check out follow us on Blue Sky for NWB and DANDI. We have YouTube channels where we'll post videos like this. You'll see lots of talks that we try to put up on YouTube as much as we can. And yeah, with that, I'll just thank our supporters. Brain Initiative, the Kavli Foundation, Simons Foundation, and Michael J. Fox Foundation have all been very supportive of this. And yeah, that's it. Oh, one final thing is we have a survey. It should take you like 3 minutes. You can probably do it during this Q&A that's about to happen. You can either go to this URL or take a picture with this QR code. It really helps us out a lot to understand to help us do better in giving these types of presentations and understand who all of you are. Okay, and with that, I'll take questions. Oh yeah, please use the mics if you want to ask any questions. Olivia, yeah.
How did you generate all of the code that was detailing all of the data sets you can read from NWB? You were on the website and you clicked the button that generated all of this code. Can you show this again? Because I think that was very useful.
Oh, Neurosift, yeah. Right. So this Neurosift app, it serves many purposes. It provides the interactive visualizations, but if you scroll down, there's this maybe perhaps a little bit hidden tab, which we're working on making it more active because I think it's very useful, that basically it programmatically inspects the NWB file and then generates the Python code to read like just about every data element. And because we've heard feedback that it can be difficult to kind of dig in, you know, these NWB files, the thing about NWB is it needs to handle like any neurophysiology experiment, and so because of that, it's quite complex. And because of that, it kind of can be a little bit of code to get to the data elements that you're looking for. So our way of dealing with that is just generating this code, so you just got everything there. So this can be used for a human, or it could be used for an AI, right? So you can plug this directly in to any AI and be like, here's how you access anything in this NWB file, and then, because otherwise the AI will do a lot of like iterations, like trying out ways to access data that don't work, and then it's just you're losing a lot of cycles. This makes it super easy. Yeah, Tomo.
This is a microphone. It's a great way to ask questions so that the whole audience and the recording can hear. And as a comment, I have a question. Yeah, going along those lines, what are you guys thinking? And is there like a highly recommended way? Do you guys prefer to have, if you guys already have an MCP that is well established, perhaps some skills that you kick over to our agents, or even a agents.md file that already has some of this baked in versus like deferring to you guys to generate the code for us?
Right.
Let's say that we're scientists and computational scientists in 2026, and we have our agent harness of preference. What is the best way, and what are you guys thinking in terms of providing a agentic friendly way to get into this? And in particular, I urge you to put yourselves in the shoes of the many a graduate student or postdocs here who like are entering the data deposition stage of their projects and are now being tasked by their PI to, okay, now convert all your data into NWB real quick because we had the data sharing thing on our grant.
I get
How do you
it.
save them out?
I get it. So our first step towards this is that chatbot. And that is an agentic framework that works on finding the relevant documentation and presenting that to you. Because I think one of the challenges is we just have so much documentation it can be difficult to navigate, and that chatbot makes it really easy. So and it also has the technology where it cites the source, so you don't have to worry about it like hallucinating. You can actually go and confirm that that's truthful information. And that, I think, really does help a lot with conversion, but it doesn't like hook into an agentic framework harness, as you said. We are absolutely working on that. Like, we have an active we're actively developing a Claude Code skill for NWB conversion. Though it does, like in testing, it works 80% of the time. But the thing is, we can't trust that it's going to always make the right decisions. And so we're really in the process now of like validating that and making sure that we can like catch as many edge cases as possible. But yeah, we're really thinking about how we can leverage on the data conversion side and also on the data analysis side. It's like, how can we develop a skill that has the guidance and the tools to call the search APIs? So you can just say, oh, this is my scientific question. Is there a data set on DANDI that's relevant for it? That's another type of agent and framework that we're building. Along with how you would ingest this data into tools like Pineapple for further analysis. So basically, like the whole pipeline of like conversion, annotation, search, and analysis, we're thinking about how we can integrate AI, and we just like don't have anything to release right now, but we are definitely actively working on it. Oh yes, please.
Hi, thank you for that talk. So speaking of sort of integration with AI and that paper that you showed about Poyo using NWB to do some sort of model-based inference, some file formats, especially with image data, for example, are better for use with model training. Others are worse in terms of random access and things of that nature. Are there any sort of considerations built directly into NWB to facilitate model training and inference, or is that something that hasn't been thought about yet or hasn't come up?
Yeah. So is this more about like the data layout or I guess, okay, let me back, sorry? Yeah, okay, so let me, I guess, back up a bit. When you're talking about training machine learning models in a general use case, there's like kind of two things you need to worry about. One is like sufficient metadata to understand like whether this data would be relevant for you and what it means. And then there's also like efficient data layout, efficient data loaders so that it's performant, right? So the NWB schema handles the metadata aspect, and we're filling holes where they appear. The HED integration was somewhat inspired by a machine learning application where they were just like, we don't know which NWB data sets to look at, and it's like the HED is intended to solve that problem. So that's one direction. The other is data layout, where we've actually done quite a bit of work on making this data efficient to load and to read. So HDF5 provides really powerful chunking and compression tools, which I guess we cut for time, but I guess we could have talked about that. But that allows us to represent data in a way that we can use lossless compression on individual chunks, which allows you to store it efficiently and still read it efficiently, essentially. And then for cloud compute, we have two approaches. One is to offer ZAR as a possible backend, which was intended specifically for optimized for cloud compute. Another option is Lindy, which basically like indexes the HDF files and HDF5 files and allows you to read them as if they were ZAR. And actually that, minus the indexing step, once you've done that, it works quite efficiently, just as efficiently as ZAR. So those are the technologies we have. And right now it's a matter of iterating with the people using these technologies to understand, okay, what are the compression and chunking and annotation settings that we need for these applications? And that's where we really want to work actively with the community, because I think we have the tools in place. It's just a matter of applying them in the best way. But thank you. Yeah, it's really something we're thinking a lot about. All right, great questions. Yeah, thank you all. Appreciate it.
All right, well, we're a little early. We're going to start getting set up for our next session here, but we'll get started at in 10 minutes or so. Is that good? Cool. All right, well, reconvene in 9 minutes. Plug in. All right. Hello, everyone. Okay. We're going to go ahead and get started. So we're now going to get going with the IBL Open Data Platform and Tools. So join me in welcoming Olivier Winter to the stage.
Hi everyone, so that's the last tutorial of this session. It's been a long session. Today we are going to do things a bit different than what we did last year, and there's going to be two main changes. The first one is that we are going to present scientific results first and then go over the tutorial that explains those results. The second change is that we will have a tutorial to show how to load the data with the IBL tools, but also with DANDI, because we have all of our data sets on DANDI sets, and that will be one of the other differences for those that attended those tutorials last year. So for this, I'm going to get the help of Michael Schartner here. He's going to present his work. He's the data scientist at IBL. I'm Olivier Winter. I'm an engineer at IBL. So the two of us, we are going to have a complementary way to present, to go through these results. So how this is going to unfold? First, we're going to go over the scientific background. So we're going to discuss a bit the relationship, or more the absence of apparent relationship between function and anatomy with the IBL datasets. So it's pushing a bit further from the original brain-wide map paper. Wow. And to illustrate a bit this in terms of tutorial, we'll have two parts of coding. One part, we are going to concentrate on looking at computing those functional vectors or functional features of single neurons for a single insertion. And we're going to go over this with both the DANDI set and both the IBL data sets. And then you will have to imagine that we're going to scale up and do this for all of the IBL recordings. And then we're going to work with the data that we have prepared for all of the IBL recordings with all of those features to do some kind of large-scale analysis. So that's how this is going to go for today. Normally at the end, you would have a feel on how we organize some kind of large-scale processing to achieve those kind of scientific results. So to start with, to kick off with the scientific background, I'm going to get Michael to present a bit his work on those distribution processing loops.
Okay, hi everyone. Hope you can hear me okay. So we have always these huge data sets, right? And as you heard before, there's so many different data sets. And how can you actually combine all that and make good use of it and say something truly new, given that you have these massive new data sets? So in IBL, we have this brain-wide map data set, which is all based on a standardized forced binary choice task that you see sketched here in the cartoon with the mice on the left. And there's trial structure. So the animals see a stimulus coming up on a screen either on the left or the right side, and then have time to react to that and need to move a little wheel to put the stimulus in the middle, and if they get it right, they get a reward. So a very simple task, but we try to do it in a distributed team science way where many labs try to put pixel probes at different locations in the mouse brain. So, and as you see here again, a bit more on the left side, in this plot, these trials are kind of in blocks. So, you know, the probability of a stimulus being left or right side is fixed for a bunch of consecutive trials. And on the bottom, you have a little bit, again, sort of the timing of the task. You know, they need to be still, these animals, before they react. And then they get a reward if they put the wheel in the right place. And on the right side, you see, you know, a bunch of behavioral traces. We also keep such as wheel velocity and video based time series such as whisking and licking using deep lab cut back in the day, but now we use lightning pose, which you may also have heard of. And at the bottom right, you see example raster of a bunch of neuropixel recordings that illustrate a bit the neural activity during this trial structure. So that's the simple task that we have, and here you see it illustrated in a video. So left is kind of the setup, where the mouse is in this, you know, the striped mouse holder. You see the animal sitting there and seeing the task. And on the right, the brain, you see trial average activity across the whole brain-wide map data set, like 60K neurons after spike sorting and QC, and you see how they light up when the animal reacts and moves, for instance. In particular, the whole brain is active after feedback. So, and here's a sketch just to illustrate again, you know, the density of all these neuropixel recordings across the cortex. So, if you squint a bit on the top, you see kind of the location of lots of probes. So it's just, you know, the one hemisphere is densely pierced by all these neuropixel probes. Of course, not all at once. So per animal 2. And on the right side, the big colorful map there is a 2D representation of all these brain areas. So it's called a Swanson map, which we use to map the 3D brain locations into 2D, so we can have an overview of what area may correlate with behavior. So here again, a bit the data, just to see a bit the numbers of this big IBL brain-wide map data set we're very proud of. It's more than, it's around 700 of these neuropixel probe insertions, and there were 12 labs trying to do this independently. So that's one of the big team science advances we had in IBL, right? That we had like all these labs in different countries trying to do the same thing independently, and we were developing standards for analysis and data along the way. So what can you say now about all that, right? So the point of all it is, of course, to say how can we say something new about the mechanism from perception to action? That's sort of what most of the systems neuroscience with mice is about. And as we show in this paper here, we have, for instance, these neural correlates for the stimulus side. You know, on top you see a bunch of these Swanson maps, these flat maps where you have brain regions colored by, let's say, the correlation with a particular thing. The variable here is the stimulus side, and we have different methods to correlate behavior with neural activities. For instance, in panel A, so top left there, we have a decoding analysis where you just decode how can we decode the stimulus side from a brain area. And then there are different ones. So for instance, in C, we have one we call population trajectory, which is using trial-averaged PTHs to see kind of how different they are for in different brain regions for stimulus right or left. And in the bottom, you know, we try to summarize sort of the correlation of the stimulus side with all these different brain areas. And in particular, a row is a different analysis method, and if all these rows overlap, then the methods agree. So, and we did so with a bunch of variables like choice, stimulus side, feedback type, and one of the interesting observations was, so if you have to squint even more for this little SI figure, you see in each row one of these task variables, stimulus, choice, feedback, and there's also wheel speed and wheel velocity. And then the x-axis is all these brain areas, and one of these bars is kind of the average across sessions. So each dot is a session, right? We have lots of sessions, lots of animals where we recorded this brain area. And you see there's a lot of variance across these areas, and there's a lot of variance across sessions as well. So the upshot was a little bit of this whole broad analysis is every variable everywhere in the brain. And now that we have this data set, we can look at this a bit more closely. And we do so, you know, by a second wave of now papers that tries to analyze this data set. And hopefully some of you will also have a look at it. Downloading it and analyzing it via DANDI maybe is a very clean way, but of course we also have our own servers where it's stored and lots of own documentation that you can throw in your favorite agent, LLM agent that can explain how to access this stuff and how to make sense of it. And here this method is now a closer look at the data set by using trial averaged activity. So, you know, in A, you have a bit the hypothesis that each brain area is functionally specific and whatever visual cortex is only doing vision. But on the other hand, there's of course the hypothesis that all brain areas do a little bit of everything, and we just need to quantify to what extent that mixed selectivity is not homogeneous. So in D, you see what you can just do by trial averaging different PTHs, right? So we exploit this IBL task structure by looking at these different events and defining a few window sizes, you know, like PTH and PSTH. So it's actually the same thing, right? One is peristimulus time histogram, the other one peri-event time histogram, so whatever your alignment event is. And in D, you see if we just look a bit at these the average across all 60,000 brain-wide map cells, so how does it look like? You see, for instance, that averages the activity is very high after feedback onset and after movement onset, which is sort of in line also with the video I just showed at the beginning. You know, everything lighting up when the animal moves. And in E, you can say, okay, so for one neuron, if we just concatenate a bunch of PTHs that somewhat exploit the trial structure as much as possible, what does it look like? And there's a lot of interesting response patterns. So an F, like on the side there, you see a lot of very different neural responses. So a little bit what Ben also highlighted in you know, in one of his web pages. It's very interesting to how easy you can just by trial averaging see a crazy variety and crazy diversity of neural responses. And you know, some are, for instance, only active here for the stimulus onset, depending on, you know, all these little S's is here stimulus aligned PTHs, and all the little M's is kind of movement aligned PTHs. But others are also super active for feedback, be it positive or negative, and then there are various combinations in between. So the question is, are all these, you know, stimulus responsive neurons, are they primarily sitting in a primary visual area, or are they all over the place? So that's a bit the hypothesis that we try to illustrate today. And in particular, this is a very straightforward method, right? You just concatenate a bunch of PTHs, and like this, you have a functional feature vector for each cell. And if you have exactly the same length of this feature vector for each cell, you can just combine the whole data set, right? All 60K cells, very standardized, and then you can try to say something about the whole data set as a whole collectively. And for instance, here are a bunch of example cells. If you just first do a k-means, so you know, you look at the big space of all these different responses across cells, and you cluster it in a bunch of clusters, say 25 clusters, and then you pick a few randomly, just to illustrate what this functional cluster looks like. Then you see, you know, this cluster, for instance, are all these stimulus responsive cells, and then you look per cell, where is it sitting, right? So we have the histology in IBL. So for each cell, we have an X, Y, Z position, and we also have a brain area. And so you see already here that, you know, these very sensory sensitive cells are all over the place. There are a bunch of auditory ones, a bunch of visual ones, you know, a bunch of midbrain ones. And so how can we just illustrate it all in one big plot? So that's kind of what we try to make you guys try today, maybe, or show you, you know, as a way to combine all this data set across all these many animals. And here, this colorful one in the middle, on the y-axis, you have the 60,000 cells, and each row in this image is such a response vector, you know, for the cell. And we group it, we order that by k-means. So we just put it into 25 clusters. And for instance, here again, you know, the little blue one here, cluster 20, was the very sensory active one that I highlighted a bunch of times before. But the other ones also, you know, show very distinct and interesting average response patterns if you force all these neurons into response groups. And you also can already, you know, appreciate in this image that you get a really nice one picture overview of all these different response types across the data set. Now, if you order that same image just by anatomical region, so here in this case it's just dividing up the one hemisphere into 10 cosmos regions. It's kind of this very coarse parcellation of the brain, right? So you could expect that, okay, is it very different, the response patterns? And not really. Like with this metric, you just see, okay, you don't see this diversity in responses if you just divide it up anatomically. It all looks pretty much similar. And on the right side, you just see the average per big cosmos brain areas. You know, isocortex, for instance, this green one here, is the same as there. Okay, so if you do another thing, another embedding, there's by Carson and others. They developed an embedding algorithm called raster map. You may have heard raster map. Raster map is just a 1D embedding that puts together similar response vectors next to each other. And then you get this picture, which is quite interesting. It may not capture as many different response types as you saw in the k-means sorting of the same image, but the interesting bit here is that it puts together similar ones. So you may see sequences, for instance. If you squint a bit, you may see very faint sequences, and here you have these very strong sequences. So that gives you a little bit gives you a bit of a hint of how signals are traveling. Yeah. So, and the interesting bit is now if you do the same raster map sorting, but just for one brain region, right? So instead of having 60,000 cells, kind of all of them, you just take 2,000 or so for one brain region only. And the interesting bit is you still find pretty much the same diversity of response patterns, right? So the main upshot is in every brain area you can find any of those big response types. So that's quite nice. And so another way to visualize that sort of hunch we got from the raster map embedding is that if you throw, let's say, all the 60K cells here on the left side in a 2D embedding of UMAP and color it by anatomical brain area, then you see not so much of a pattern. Maybe if you squint a bit pinkish one side and greenish is more the other side. The cortical ones is kind of green and pink is more the midbrain stuff, as you see here in the actual 3D brain location of all these probes. Here all these neuropixel probes, all these lines. That's actual brain 3D space. And of course here the anatomical coloring makes a lot of sense by design. But conversely, if you color that functional UMAP thing by 25 k-means clusters, you see a pretty nice parcellation, partitioning it up. But then once again, if you use this color coding of your functional groups in the actual XYZ brain space, it's salt and pepper and all over the place. So there's not so much of a clean link between anatomy and function, and it seems to be interconnected processing loops that dominate even these most basic IBL variables such as choice, feedback, and stimulus side. And with this, I hand it back to Olivier that gives you a bit of an overview how to access the data and reproduce these plots.
Thanks, Michael. Okay, so we've seen a bit what we want to achieve, what we're going to do now. So we're going to scale back and go back to the beginning. So here is, if you want to follow along using a Colab notebook, here is a link, a QR code. And what I enjoin you to do is that before I introduce what the technical parts of what we are doing, you can try to run the first few cells, okay? If you want to, because it's going to take a few minutes to install the environment and download the data. If you want to modify the code yourself, you may want to make a copy first of the Colab notebook in your own Google account so that you will be able to modify the cells and run it yourself. So I want really to emphasize 2 points in this technical. The first one is going to be data management. And how are we going to query all of those data sets? So I'm going to explain this and the difference between metadata and data. The second thing that is really important is here we are going to do a very simple analysis, computing the PETHs for each of the clusters, and then we're going to concatenate them together. We call this in computing a map-reduce architecture. We're going to map this process to all of our insertions and then reduce this to get a final matrix. We chose this example because it's trivial in this case. There is no relationship between any insertion, so every compute insertion could be independent, and then at the end you reduce all of them together in the same raster map. But this is really something I want you to take away with you today, is this kind of, when you start working at this scale, you have to map processes at a small scale, and you have to find how you're going to aggregate this into a main data structure. Okay, so first the data. What is happening when we are going to query the data here?
what's happening is that at IBL we decided to put the metadata in a separate database. Why? Because if you ask about people that are data managers, they're going to tell you the hardest thing to manage is the metadata. It's much smaller, but it's very complicated, and we decided to store this apart from the actual raw data. So what people do when they want to query the IBL internally and externally, you do a query to this metadatabase that is a web application. And it contains everything from subjects, procedure, genomics, the brain regions, and your query is going to send you back some relevant dataset links. And with those links, you're going to hit one of the bulk data, the raw data repositories that contain all of the IBL data, the spike trains, but also the raw data. And here we have several backends to load this data. We have our own AWS bucket, which is, as Ben was saying earlier, is something that Amazon provides us for free. We also have our database, our datasets on DANDI, and we have our master copy that is on the Flatiron Compute Center. So this is what is going to happen when we are going to make the queries. First, we do the query with ONE, we find the relevant datasets to load, and then it's going to hit the backend, the raw data, the large dataset files. Okay. Ooh, it's not private, but part one. So normally to run because this one? Ah oui. Anyone with the link? Okay. Let me check the other ones, because I'm sure. Anyone with the link? Okay. So yeah, split those notebooks, and they didn't carry over the permissions. You should be okay. Are you okay now? Yes. Sorry. Okay. Well, that's good, because we are going to switch to the notebook anyway. How is the size of those? Okay. So normally, if you run the first few cells, on the first cells we install the environment. Then we are going to set up this API that we call ONE. And this API is going to be the thing that is going to negotiate and discuss with the metadatabase. Okay. The last thing we are going to instantiate is what we call the brain region object. So this object contains all of the ALEN Atlas brain regions, so that we can start working with understanding where we are in the brain and going through the hierarchy of regions. Okay. The second step is we're going to download the datasets. So in this case, we're going to select a PID, and I'm enjoining you to click on this to run this cell now, while I'm going to explain how you get those probe ID or experiment IDs. In this case, we load a spike sorting loader. The reason why we have those kind of loaders is that we have several revisions of spike sorting. You have to bear in mind that IBL is a project that spans 6-7 years. And over those 6-7 years, a lot of the methods have been evolving. So if people have published papers with the early version of the spike sorting, we want to maintain the compatibility with those papers, but also we want the new users to have access to the latest spike sorting. And here we are going to load the spike trains, the clusters, the cells and the channels, and we are also going to load the trials. So while this is running, I'm going to show you how to decide how to query. And the easiest way to query is to go to this visualization website. You have the link here. And the idea here is that you can select any probe of the brain-wide map and look at where they are in the brain. And you can also look at what are their responses down there to the task. So I'm going to go all the way down to the FIS. You have many displays that show the quality, that allow you to assess the quality and the response of the cells to the behavior. And here we are going to have to focus on this display particularly, where I have the list of cells that have been acquired, and we see raster plots on panels B, C, D, E, and F. And those raster plots are individual trials, so each line is a trial, aligned to a specific event in the task, and they are sorted by different metrics in the task. So typically, here we are looking at the contrast. On this example, we are looking at the side and of the response. And in this case, we are looking at the answer, if it's correct or incorrect answer. And here we could look at each neuron individually to see how it changes the PSTH. So the query part is done here, and you have the query names, how to run the query. So for example, here I'm going to query an obscure region that is in PARN. I'm going to select this. It looks like we don't have internet anymore. All right. Well, for the purpose of this, seems I've lost the internet connection here, so I cannot refresh this page. Bon. Ouais. So the idea is that you'll be able to look up for a probe insertion according to the brain region. And this is what I did here. I wanted to look at one probe insertion in particular, and after you've queried for this, nothing anymore. After you queried for this probe, you can copy-paste this probe ID that will send you, this is something that you can query after. It's the insertion ID, and it uniquely maps to the recording that concerns this query. So once we've done that, we have downloaded the spike sorting for this specific recording that we selected. We're going to select the clusters. So here we want to do the analysis in the interest of compute time. We want to do the analysis only on the cells that pass the quality metrics. So we compute a set of quality metrics as IBL, and this is the selection, the query that is going to select only the good cluster, the good cells from those quality metrics. So in this case, if I select the good cells for this recording, I get a table of 80 cells that are identified as passing all of the quality metrics of the IBL. So this is the cells with which we are going to do the analysis today. So once we have selected those 80 cells, we want to compute the PETHs. So here we are going to, as Michael showed, we're going to compute a set of 20 PETHs for all of the cells, all of those 80 cells. And those PETHs are defined uniquely by the alignment event in the task. So we need to know what is the time zero and around which event we are going to align. They are also going to be defined by what is the selection of trial we are going to keep. For example, the probability of the block, the response type, if the response was correct. So for each one of those, we are going to run this query. So here we've written some helper functions to do this. So here, this is the code that translates the table you see up there into parameters. And we are going to use those parameters to align to the PSTH. So for example, this block L is going to align on the steam on time event. 0.4 seconds before and 0.1 second before, that's the time window relative to the steam on time. And I'm going only to sum the trials for which the probability of the block was 80%, the probability left of the block. This is the corresponding, the reciprocal query, exact same query with the probability left 20%. Okay. So this, we're not going to go over this full code, but what I want you to take away with you is that here we are not going to loop over the clusters. I'm going to loop over those PETHs. We're going to loop 21 times to compute those, and we're going to do PETHs in 2D, which I mean we are going to do all of the PETHs for all of the neurons in one go. And now usually it's very fast. This is going to run in a few seconds. And now what we have all of the PETHs have been concatenated together in a big array that is the number of cells in the row and the number of time points, number of columns. So now we're going to display this as a function of probe depth. So the display we want to have is a display that is very similar to what Michael has shown. So we have our PETHs for 80 neurons, with each of the individual PETHs denoted by the vertical lines. And we can see a bit the response of each of the neurons as a function of the depth at which they were recorded on this insertion. So here on the right, we have the column of the histology column for which this insertion went through. And if I can find it now. This one. So it's at the back of the brain. And it's going to be this insertion you see in red over behind, and it goes through the cerebellum. So it's really at the back at the back of the brain. Okay, and we see the histology column is the same as what we are looking here, and we see our neurons here, which are the same that we are looking at. Okay. So how does it work? There is one thing here, so it's conventional plotting code. One thing that is interesting to note is to get the histology column, we have quite useful tools, which is the IBL Atlas, that are able to navigate those regions. It's the exact, it's a backend to the ALAN Atlas, and that allows you to do those kind of displays to plot the histology column so that you always know in which brain area we are supposed to be. So that's the part of that's the IBL way of doing this, if you want to use the infrastructure of the IBL, but you have the exact same way to do it with DANDI. So I'm going to show a bit the DANDI way to load this data. So in DANDI, you would be doing more or less the same exercise going in the Viz website, but you can also use the framework that Ben showed a bit earlier. This Atlas website, for which for all of the brain-wide map, the DANDI set 409, which is IBL, you can go and navigate all of the subjects. So here I've selected this specific subject, this specific recording. So here the data is per session. So in a session, you can have multiple recordings if you have multiple probes, okay? So for example, some people can use up to 6-7 neuropixel probes. In this case, we would have 6-7 recordings within the same session. So here we are looking at this session, and it turns out that this insertion that we are looking at has been recorded simultaneously with this second insertion that is up there. So that's really a cool tool. Huh? Thanks, Ben. Yeah. So that's also a way to query the data per brain region if you're interested in looking at some probes. So once you have the information, you need two informations here. You need the session ID and the probe label. So here it's the probe 00. So all of this code is going to get the DANDI set. You instantiate the DANDI API client. This is a kind of boilerplate. You get the client, you look at the assets, and you look at the processed files. One thing that is useful to know about the IBL DANDI sets is that we decided to split the NWB files in separate files. One is the raw data, one is the video, and one is the processed data. The reason is that in order to make things easier for people that want to do analysis, you don't want to download always the raw electrophysiology data. The only thing you want to do is to compute PETHs on spike trains. So here we are going to have 3 assets that shows up. But here I just want to get the processed file that contains the behavior, the spike sorting, the video segmentation, and the pose estimation results. So once we've loaded this processed file, so here we download the file on this. So normally it takes 1 minute from DANDI to Collab to get this file. And here I decided I wanted to give a shout out to Pineapple. I find it very easy to load an NWB file with Pineapple. So to me, that's what I chose to do. It's just to instantiate Pineapple and load the NWB file. I do my unit selection here. So the same as we saw before. I just want to select the units that pass the quality control. And then after, I did want to reuse the code that Michael wrote. And we have a different way to serve the spike trains. At IBL, we serve a single spike train that contains all of the units. And the spike train is ordered by time. So they all interleave the units together, while in Pineapple and in NWB, the spike trains are per unit. So in this case, since I want to reuse the same code, I just don't want to change the analysis code. I'm going to recreate the spike trains as we used in IBL. Why am I saying that and showing this to you? It's boilerplate, but there is a design principle that could be nice, is you keep the analysis code separate from the loading, and you make sure the loading gets the exact same input that your analysis code needs. And this is what I'm doing here. That allows to decouple the input-output from the actual analysis code. Once I've done that, I can rerun the exact same code as we've seen before, those 21, and I'm going to display the result in the exact same manner. And I will have the exact same output. So the brain regions, the trials, it could be slightly different, the way it's shown in DANDI and in IBL, but it took with a good agent and 30 minutes, it was easy to convert the loading functions for this to reproduce this notebook using DANDI. So here you have two examples, one IBL way to load things, DANDI way to load things, but all of the analysis downstream is the same. So that will conclude this first part. So we did for a single insertion, we pulled all of the good neurons. We computed plenty of PATHs, 21 of them. We looked at them. So here we can see that we're in the cerebellum. We can see that it looks to me, correct me if I'm wrong, Michael, but there is some lateralization going on here. Like for example, this is left and right side. We can see that this is much higher activity than on the left side. So now you have to imagine that we're going to do this for all of the insertion, rinse and repeat. And we're going to loop over 700 insertions and do the same processing. So it's embarrassingly parallel. It means that it's trivial. In this case, we can just launch the same process in different machines or however we want to do it because they are completely independent. Once we have all of those responses for all of those cells, we're going to concatenate together into a big matrix. So that's going to be the second part of the tutorial, is that we're going to load those pre-computed PETHs for 60,000 neurons. And we're going to go through a bit the figures that Michael showed. We'll display a subset of the neurons and display of the PETHs ordered by anatomy and k-means. And then we're going to look at the UMaps and display the UMaps according to those matrices. So it's the same here if you want to start running as soon as possible. The reason is that it takes a bit of time to download those two data sets. So the data sets are the concatenated PETHs. So it's a big matrix, and the matrix size is 60,000 per 1,800. It's going to download them. It's going to take probably 2 minutes if you start doing this. So here we are mostly going to work outside of the IBL environment. We don't need to query the database anymore because what we did summarize the data in such a way that now we can start launching some unsupervised and learning methods onto this matrix. So this is going to be reusing the library here that we've cloned. And I'm going to show a bit how to plot some example neurons. So those are all of the functions from Michael. So those are the same, it's not exactly the same, but that's a set of example neurons that for one cluster, for one K-mean cluster. And those example neurons show a very similar response to the task events, a bit regardless of where they are in the brain. So next, we can do this raster map and order by K-means cluster identity. So there are two displays that will come out here. The first display is going to be the average K-mean, as we saw. So those are all of the 24 clusters. And you can see that this is the typical canonical response to the task events, the functional response to the task events for this K-mean cluster. And this is the same raster map corresponding to all of the individual traces. And what's really interesting here is that depending on how you sort this, how our human eyes are going to make sense of those traces that are always the same data, but ordered in a different manner. So now we're going to do the similarity based on ordering of this raster map. So this is using a raster map, which is a library developed by Carson Stringer. And this is the order that is going. So for all of the brain map, for all of the 60,000 neurons, this is going to be the kind of responses that we have, as Michael showed. And we can do the same now. We are going to resort this per anatomical region. So we have a parcellation of the region that we call at several levels. We have the original Allen parcellation, and we can go up the hierarchy to a higher level where we think this is the level at which it's nice to do some neural analysis. We call this parcellation Beryl, that contains something like 200 regions, compared to something like 1,000 regions on the original Allen map. And we have a higher parcellation level yet, that is one of the highest level, where we have only 12 to 13 regions. And this is the parcellation level we are looking at here. So here it splits the brain in 12 different regions, which is cerebellum, cortical subplate, the hippocampus, for example, the isocortex. So you can see the response for those very big brain regions. In theory, we could split this per much smaller nuclei. So this is the same. So we have the average response per region. As Michael was pointing out, we don't have a lot of difference. We know the cortical subplate, we don't have a lot of representation there, and it's quite noisy. It's the same for hypothalamus. If you can see that the size of those regions, a number of neurons there is very small, so it's probably a bit noisy as well. But it's very hard to see some structure in there. It seems like everything is represented everywhere. Yeah. The next and last part of the tutorial, so this has been generated by AI. I had to change a bit the backend in order for this to run in a notebook. So we use Plotly for this, and kind of reproducing the fact that we are going to look at unsupervised clustering of those feature response vectors. So here you can imagine you run a new map on this matrix, and you say I want to reduce those 60,000 neurons times 1,800 time points just to 2 or 3 dimensions for those 60 neurons, and I want to look at where they show up in this unsupervised map. This is independent from the K-means clustering. Okay. So here we have a 2D, it's actually 2D UMAP of this. So that's a dimensionality reduction of all of this on 2D, on which we are going to color the dots with the K-means, with functional response, and with the anatomy. So I'm showing the... bah oui. But that's an old version of this tutorial, or what? think there has been an internet issue. Bon. It's probably going to take a while. Sorry, I should have saved a static version of this notebook, because I think it didn't save this. But the bottom line is that if you do this, you will have in the notebook an interactive way to interact with those traces, and you will have exactly what we showed before, which is the UMAP on a 2D. And if you label this by anatomy, you have something that is really that doesn't show a lot of structure. Or you can see a bit with if you really try to nitpick. But as in the 3D real space, it's really well organized, while in the UMAP 2D, you don't see the functional response showing up too much, the anatomy showing clusters on the functional responses. While, of course, if you label this with the k-means, which is kind of another unsupervised way to look at this, you will have a response on the UMAP that is much more clustered. In the right part, you can see that this has absolutely no structure. Yeah, I'm a bit disappointed that I've lost those plots, because they're quite cool of interacting with in Collab. Yeah, seems that it didn't save this morning. Sorry. Ouais. So that will conclude this tutorial. I'll make sure that this completes. I think it will take like 5 minutes, so I don't want to put you through this. No. So what did we show you? We showed you some kind of interesting scientific results that go a bit beyond what we were doing at the brain-wide map. What was really important, what I really wanted to show is how to map a simple analysis that you do on a single insertion. You can do this with DANDI, or you can do this with the IBL way to do this. And the last part is that you map this part on all of the recordings, and then you reduce this to a big matrix on which you can do unsupervised analysis and try to understand things at scale about the brain. So it's really those two things I wanted to show you, the single session part that you can look into this website, looking at the single neurons, which is really important way to do things. But also, once you have an hypothesis of how to take this up to the next level and to look at 60,000 neurons all over the brain at once. So that was the object of this tutorial, and I will be ready to take any question with Michael, and thank you.
Please make it orderly line, not everyone at once.
So maybe I have a comment on the previous question. There was a question about how to prepare the data for large models. That's a very good question, because I think, to me, the NWB is a really good archive format. But what we see for analysis, and that's an example here, is that you may want to reformat or to summarize your data for your specific use case. So typically for the large neural model, you may not want to use the LFP at the sampling rate at which it is. You may want to resample your LFP, you may want to resample your wheel, and all of this, it's kind of, you may not want to save this data. So you will have to do some kind of pre-processing and what we call staging the data first, for use of those large neural models. So I think there are some initiatives about this, and the large neural model providers are also coming with the tools to prepare the data to input into those models.
All right, I have a question, and it's basically the question that Talmo asked me, mostly because I'm trying to learn from my peers and figure out how best to do this. So have you been thinking about preparing this data for like operating with agents specifically? Like, are there skills or other types of frameworks that can facilitate agentic data analysis?
Yeah, absolutely. I think we are all working on those kind of solutions. Right now, it's not ready yet to be shown. But the ultimate dream is to have an agent that you can set loose on DANDI, and that is going to make cool hypotheses and cool results. Yeah. So yes, we are actively working on it as well. We have our agents. We have a set of Cloud.ai files, Codex MD files, and yes, we certainly have people working on that.
I feel like we all need to meet and discuss and share notes.
Yeah.
Yeah.
Yeah, it's like our job is really changing in the sense that now it's really about how to write the objective functions, how to really write the thing that is going to tell you that your agent comes up with the right answer. That becomes our job more and more.
Just one comment. I mean, that's the beautiful thing about open data and open science, that you have all these repositories with tons of data, tons of documentation, and that's all machine readable. So you can throw your favorite agents at it, and now is the gold digging time to find some nice results with open data. So I hope you all got inspired and have a look at all these data sets and make good use of it.
Back at 1 o'clock for the main Cosyne tutorial.
Thank you.
Yeah. Thanks, guys. Yeah, great work. Oh, okay. Small logistical announcement. We love all of you guys, but we need you to leave and exit the room and go back in through the door.
Yeah, so if you can exit all the way to where you grabbed your badge from, because we need to sign people back in for the next